So here we go. You already drew early prophase 1. This is, by the way, of this chapter, this is kind of the day you want to really have your A game, or you'll be swimming a little bit as far as what's going on in meiosis. Okay, so you've got this handout that I gave you, but it doesn't have everything. You're going to want to write some notes below each thing on the handout. So go ahead and fill in what's missing from what you drew yesterday under the picture. Remote folks, it's up to you. If you want to reproduce these drawings yourself, you can. I'll also post the handout for you if you want to print it out and write your own notes like the kids in class are doing. So early prophase 1 looks an awful lot like prophase in mitosis. It's really not different. Different thing is we're following a little more detail. We're noticing that there's pairs of chromosomes that have similar colors and sizes. We're following a couple of genes. We're following, since this is a fruit fly, fruit flies have 8 chromosomes. We're following the wing shape gene, and this fruit fly happens to be heterozygous. It's got one big W and one little w. We're also following the eye color gene, and this fruit fly is heterozygous for that one as well. Riley, you had a question.
What would you write the name, like early prophase 1, or do you want us to write meiosis 1? I like the top of like our name.
I would do both.
Okay.
Yeah, I mean, fill in meiosis 1 where it says A, but then underneath put early prophase 1. That's the name of the phase. Meiosis is divided, like subdivided into two sections, I guess. There's meiosis 1 and meiosis 2. And it's the names will be familiar. It'll be prophase 1, metaphase 1, anaphase 1, telophase 1. And by that time, you'll have 2 cells, just like you did in mitosis. But then meiosis 2 takes those 2 cells and divides them as well. So you're going to wind up with 4 cells. That's how this works. So this looks just like, we've got the nucleus dissolving, we've got the spindle forming, the chromosomes have formed. But this, honestly, give me like solid absorption of information and concepts for this and the next phases, and you'll be set for the rest, because this is the only place anything weird happens. And what's weird in meiosis, what's different from mitosis, is the very next thing here. Instead of these guys going straight from this to lining up single file down the middle like they did in mitosis, you remember that, right? What happens instead is they take a minute and they find their partner. Every chromosome, remember, has a homologous paired chromosome. They find each other. In late prophase 1, homologous chromosomes find each other and they stick together. There's a process called synapsis. Two homologous chromosomes stuck together like they do is called a tetrad. Tetra means 4. Since these chromosomes are duplicated, right, already replicated, gone through S of interphase, we call them tetrads because it's like there's 4 chromosomes together. The first and really, really, really important thing that happens that's different from mitosis here is while these chromosomes are stuck to each other, they trade pieces. They trade pieces of each other. There we go. So I'm not a great artist with PowerPoint, but now I can maybe make this work a little bit. It'd be like if the bright red one traded little pieces with the dark red one. We'll just use black. So they end up literally being little Frankenstein chromosomes that have traded pieces. And remember, what these chromosomes are these super densely packed balls or X's of DNA, right? So what you're doing here is trading a whole bunch of alleles. You're basically taking the genetic deck that your mom and dad, because one of these chromosomes came from your mom, one of them came from your dad. You're taking that genetic deck that your parents dealt you and you're reshuffling it. You're scrambling up your mom and dad's alleles all to heck so that when you deal them out to your kids, they're going to be different. None of your kids are going to get the same genetic deck that you were playing with or that your parents were playing with. You're scrambling all it up. That's a big part of what sexual reproduction does, is create diversity like this. So it's this crossing over is thing one that makes sexually reproducing organisms different from each other. When this winds up at the end with 4 different cells, they're all going to be different from each other, partly because of the thing that just happened. Crossing over is what it's called. Okay, so crossing over is thing one, makes meiosis super special. Maybe put a box around crossing over and underline it a bunch of times and put some arrows so that you know that's a super important part of this. Questions so far? So that is prophase 1. Early prophase 1 looks just like prophase in mitosis. Late prophase 1, we've got this pairing up, the synapses happening with crossing over, trading pieces of chromosomes back and forth. Then the second thing that ensures that the offspring are going to be different, that these cells are going to be different from each other, is, ooh, that's fun. I'll go back to that. Metaphase 1, when they line up like this, they don't line up single file. Not only do they not line up single file, right? They line up in pairs. It is random which side of the cell each member of each pair winds up on. Essentially, this is like, for this fruit fly, this is like flipping 4 coins at once, right? It's a 50-50 shot whether the dark red chromosome winds up on the right or the left. It's also a 50-50 shot independently whether the dark blue chromosome winds up on the right or left. So you're doing that 4 times. It's random which chromosome of each pair winds up on which side. So it turns out, for this fruit fly, beyond the variation that crossing over already put in there, remember these are all already kind of little Frankenstein chromosomes because of crossing over, then you introduce some more randomness because there's 16 different ways these pairs of chromosomes can line up. That's 2 to the 4th, right? 16 ways. Think of it as 4 coins. You could have all heads on this side, all tails on this side. You know, on this side, it could be heads, tails, tails, tails. It could be heads, heads, tails, tails, and so on. 16 different ways these guys can line up. For you, when you have 46 instead of 8 chromosomes, it's 2 to the 23rd, which is a lot of different possibilities of the ways your pairs of chromosomes can line up. So we're talking about a lot of variation that gets created by this step, just like a lot of variation was created by crossing over. So crossing over in late prophase 1 and independent assortment, which remember we also talked about with Mendel when he figured out that traits don't affect each other. He just happened to pick the right traits that were on separate chromosomes. It's only true if the genes are on different chromosomes. But independent assortment is the fact that these guys flip independently of each other. They wind up on either side of the line independently of each other. So crossing over an independent assortment, that's what you need in your brain as far as meiosis today. That's the take home. Crossing over, independent assortment are the things that make these cells that we're producing different from each other. Beyond, past this, it just looks like mitosis times 2. Watch. Right, did you all get all that written? Okay. So next, oops. Anaphase 1. The pairs pull apart from each other. That's all, just spindle fibers pulling. And notice the difference though. This is not chromosomes being split in half. They're just moving apart from their partners. Telophase 1, the butt-shaped thing formed. Nuclear envelopes are coming back. So here we are at the end of meiosis 1. What we've got looks kind of like the original cell, except half as many chromosomes, right? We did more than that, though, didn't we? We let these guys talk to each other before they split. We let them trade parts with each other. And then we made it random which one wound up in which cell. Right? It's the dark red chromosome that wound up in this one and the bright one that wound up here, but it's the bright blue one that's here and the dark blue one that's here. Colors don't matter, but I'm just trying to show you. There's two ways this got randomized. That's meiosis 1. That's the first round of divisions. We now have 2 cells. We're shooting for 4. So here we go. Prophase 2. Nucleus goes away again. No surprises here. Everything that's coming you've seen before. Meiosis 1 was a little different because of the pairs, because of the swapping of stuff. This is just basically mitosis, all right? The nuclei disappear, the spindle forms, we've got chromosomes. Those chromosomes are about to line up, single file this time. Anaphase 2. Here's where they actually split at the centromere, right? Here's where the X's actually become little V shapes. Telophase 2. I'm not writing anything here just because I think you know what telophase is, right? There's a lot going on, but you know it all. You know the nuclear envelopes are coming back. You know the little pinchy cleft thing is forming. Or about to do cytokinesis for the second time, about to divide the membrane. But understand what we have now. These 4 cells are not just clones. These 4 cells are very genetically different from each other. We can't show all that difference very well in a PowerPoint because it's pretty complex. But like to start, we know that the dark red chromosome has got bits of bright red chromosome in it, right? And the blue has traded pieces. So that's got some bright blue in it. And it's all shuffled up. Each one of these 4 is genetically different from the other. Where is this going again? Why did we do this? You do mitosis when you want to do things like grow, right? Or heal. Or you're a single-celled organism that's real simple and you just want to reproduce. This is different. What's this for? Why do we do meiosis? Why did we do this? What are we making? Yeah. And that actually looks different depending on whether this is a male or a female. As you'd expect, we get 4 sperm cells that are genetically different from each other. If this is a male doing meiosis, if it's female, though, a little different. Turns out if it's female, we are looking at 1 egg cell and 3 not egg cells. The way female gametes develop, it actually takes the other 3 cells to kind of support the development of the egg cell. So you only get 1 egg per process of meiosis, whereas there's 4 sperm cells. All right, so this little run through, just a quick shows you crossing over, independent assortment, and the end of meiosis to where they're all different. Shows you a little better than this here, that they really do. All 4 of them ended up being different cells. Crossing over, independent assortment, 4 different haploid cells. Right.
What happens with like, I don't know the exact name for it, but with twins that are like have the same DNA?
Identical twins.
Yeah.
What happens is the sperm meets the egg, one sperm meets one egg. But now the sperm and the egg were produced by the parents with this. But identical twins separate after that. After the one new cell is formed that's different from both parents, then that cell does 1 round of mitosis, which is identical, right?
Yeah.
2 cells that are identical. Then those cells separate and each develop. Kind of a glitch, actually.
Yeah, and then I don't know what the exact scientific name for the non.
Fraternal. Fraternal is 2 different eggs, 2 different, same parents, but 2 separate meetings of egg and sperm.
Identical twins or just twins grandkids have.
The tendency to have identical twins is genetic. Yes, that does run in families. It also could be caused by fertility drugs and stuff. Okay.
How do you find girl twins that are identical?
Separate sperm, separate egg, right? One of the sperm carried the Y chromosome, one carried the X. Separate. It's fraternal twins aren't weird at all. It's just 2 fertilizations happening in the same uterus, right? Like it's just 2 separate babies. Not that identical twins are weird, just that it's a different process. Okay, good. So, find your elves. Remember your elves? Find them. I can't hear you. Okay, do you have the paper that you did with the rolling of dice and stuff like that? Do you have that digitally?
Yeah.
Yeah, find the online paper. That's really, you don't have to have the drawing right now. What you need is your dice rolling and allele picking and phenotype record of what your elf looks like. Is what you need. So we're going to take these elves a little bit further. I hope you drew them well. I hope they look nice, because they will be pairing off and making elf babies. So you want them, looking their best. We're going to dig into elf chromosomes now. We're going to say that elves have 5 pairs of chromosomes, and that each of the genes you guys rolled dice for are located in separate places on these chromosomes. And you're going to need to be writing in the alleles that your elf parent has on your elf parent's chromosomes, which is this paper I'm about to give you. It's not going to be hard, but you want to look at the example I give so you understand what's going on.
So these aren't supposed to be beaches. They should be whatever letter it says. Yep. You got it right. Just change the letters.
All right. So for example, let's say you had heterozygous for earlobes. If you had heterozygous for earlobes, here is what you would write in on your elf chromosomes. Number 1, the gene is for earlobes. Remember, it's big E or little e. You happen to have one of each if you're heterozygous. But notice there's 4 spots. There's 4 slots for earlobes. Here's how you do that. One of those chromosomes would have both big E's, Interesting. Oh yeah, look. Okay. So you'd put 2 big E's there and 2 little E's there. Let's say you were homozygous dominant for thumbs. What's the letter for thumbs? T. So if you were homozygous dominant, of course, it would be all big T's. All right. If you're homozygous recessive, for if you're for gender is different, right? It's either XX or XY. If you're female, it's X's across the board. If you're male, it's 2 X's and 2 Y's. What's the letter for dimples? Is it D? If you're heterozygous, 2 big D's, 2 little D's. You see how this works? Everybody get it? All right. All you got to do right now is transfer your parent elf's genotypes to these chromosomes.